about the soil carbon cycle. And this is the second time I'm recording this lecture. The first one was not very good, so I'm doing it again. So what we're going to cover is this mysterious process of a leaf falls and it disappears, and where does it go? And it's really quite an important topic, the carbon cycle. Carbon is organic compounds are where the energy that is fixed from the sun is captured in the form of different compounds, reduced organic compounds in plant materials. And when these materials enter into the soil, they drive all kinds of other processes as that energy flows. Those electrons are donated out of those reduced organic compounds into the soil, accepted by electron acceptors, and it just unleashes all kinds of biogeochemical cycling and growth, rising and crashing of civilizations of microbes, predation cycles on top of these growth of prey, all kinds of great, fabulous tales are had in soil because of carbon. So what we're going to cover is first a broad look at the carbon cycle, then we're going to look at plant decomposition, the first part of the carbon cycle, and look at a variety of different processes that are involved there, and then talk about organic matter formation, also called humification, which is the second part of the carbon cycle that is very specific to soil, and then end on a little bit broader implications. So the global carbon cycle, unlike the nitrogen cycle, this figure here shows you pools of different carbon compounds in different parts of the planet, and we're going to focus on the left-hand side, which is the terrestrial side of the story. And so these boxes correspond to different masses of carbon, and you can see if we compare the atmosphere to the soil, that unlike the nitrogen cycle, we have quite a bit more in the soil than we do in the atmosphere. Remember, it was quite different in the nitrogen cycle that has so much nitrogen gas making up the atmosphere. CO2, which is the main form in the atmosphere, makes up a much smaller portion of the total atmosphere. We as humans are trying to change that by pumping a lot of CO2 into the atmosphere, but we're not really making a dent in the mass of carbon so much retained there. We're definitely changing the concentration and causing all kinds of consequences of that CO2 going into the atmosphere that are associated with global warming. Anyway, let's look at some other things going on in this graph. We've got land biota. In this situation, we're referring to plants primarily, and plants equal about how much is in the atmosphere. And then we have these flows of like the carbon from the plants into soil, and these numbers on the arrows show the gigatons per year that are going to be flowing there. And we have about 110 gigatons of carbon being fixed per year by photosynthesis, and then we have the plants and the soil respiring back about how much is photosynthesized. But then we have these red arrows, which are human activities, so burning of plant material, which is still happening in rainforests that are being cleared for cultivation, and in just agricultural practices, which release some carbon into the atmosphere. Let's see what else. I think that's basically what we need to look at here. Okay, and then the concept of fossil fuels, I just want to bring this into play, is that there's a huge amount of carbon that's buried in sedimentary rocks that has been stored deep below ground out of the biosphere. But what humans do by digging up petroleum and burning it is they're releasing that carbon that was out of mind, out of the way, and bringing it into the atmosphere. And that's a huge portion of the increase in CO2 in the atmosphere. So carbon sequestration is this concept of storing some of that carbon, taking some of that carbon out of the atmosphere and getting it into the earth, into stable forms that will be sequestered there and reduce the concentrations in the atmosphere. And there's a lot of interest in using soils for carbon sequestration, and also trees. And you can see that the plants have less potential for carbon sequestration than does soil. There's also interest in pumping CO2 deep back down into the ground, into sedimentary rocks. But those are all different potentials for carbon sequestration. All right, so we're going to look at two sort of parts of the carbon cycle, as I said before. One is the decomposition of this plant material shown up in this box, the source of the soil organic matter, which is plant litter and roots, but also could be dead animals, and also, don't forget, feces that release these complex organic molecules, which we'll talk a little bit more about, that are going to decompose. And some portion of them are then going to mineralize and be converted to the mineral forms that plants need to be able to grow and grow biomass out of that carbon that they photosynthesize. And so they're going to go and make plants, and then it's going to keep the cycle going. So that's one portion, is liberating those ions, those nutrients that are tied up in the organic matter, some of which will leak out of the system. And then the other route is, after this decomposition, is the creation of soil organic matter, that magical stuff that is so characteristic uniquely to soils. The humification, this formation of this complex material that looks nothing like dead plants, that we're going to talk about later on. So we have this path A, which is about releasing these nutrients, like breaking down the plant material and releasing nutrients and energy, of course. And then B, this building up of soil organic matter. So we're going to start with A. And the first stage, remember, is that you have these big chunks of organic matter that have really high surface to volume ratios. Wait, did I say it wrong? Yes, a low surface to volume ratio. So a lot of the material is not accessible to microorganisms to directly attack the organic compounds, which of course some will. They'll hang around on the outside and work on it, or penetrate it a little bit with the fungi. But we rely on these shredders, all these organisms, to consume that organic matter and release it in smaller pieces physically, but also producing feces as well, which are inoculated bits of organic matter, which are then going to go into the soil food web. Note this, that actinomycetes get their own box, but there are no archaea. Interesting, huh? It's an old picture. Okay, so these reduced carbon compounds are the repository from energy for the sun. We already talked about that. And a major source of carbon to the soil is plant residues. And plant residues come from above, from leaves and branches and whatever else is falling out into the ground. But also carbon is coming from below ground, coming from plant roots, sloughing off, and from exudates, which are going to be a major portion of what is photosynthesized, will actually come out through the roots. And what the plant is made up of is these largely these compounds, which include these polysaccharides, cellulose and hemicellulose, which are made up of sugar units. We'll talk about them in a minute. There'll also be some protein, and there usually will be some lignin. How much lignin depends on what kind of plant material it is. The woodier it is, the more lignin it's going to have, whereas something like lettuce is going to have very little lignin. So these proportions change with the kind of plant. And you also will have a little bit of monomers like glucose, amino acids, and also some fats and waxes. So there's this mysterious thing that happens as the plant residues decompose. You start out with a whole big pile of litter. It's shredded into these smaller fragments, further broken down as microbes are consuming them, and then leaving behind this material that doesn't look at all like what it started out, and it's a much smaller biomass. So where did that material go? So this figure here shows you that. It's a really nice progression looking at these boxes, which are the total mass of carbon that initially entered the system, starting out as pure crop residue. Some portion of that, the most readily degradable stuff, will start to decompose right away, and that carbon will either convert into microbial biomass, right, because they need carbon to grow, or CO2. So boom, that's where some of that initial mass disappears, is as CO2. And what's left behind is a little less degradable. And then that material is degraded, same deal, will make some microbial biomass, some CO2, which is now adding up, right? Because we add the new amount to what was made before, and more resistant material left behind, and so on, until you get to the end where a large portion of that initial plant residue has blown off as CO2. You have some microbial biomass, but note that the microbial biomass is also turning over as we go along. And then at the very end, you have this material called soil humus. So look at the time scale. This is days, months, and then years. So decomposition is this broad term from the breakdown of organic material into its constituents and elements. And this figure here corresponds to the pie chart that we saw before, where you have a certain portion that's really degradable, but before long, that stuff's all going to be gone. And then the cellulose and hemicellulose is going to degrade next. And then the lignin is going to degrade much more slowly and over a longer period of time. And you're going to have the rise and fall of microbial biomass. You're going to make more biomass with these more readily degradable compounds, but the lignin's not going to support as much biomass. Note that the time scale is quite different for whether we're talking about being in the tropics, where maybe this whole process is going to take only 3 years to take place, the decomposition of this plant material, whereas in the Arctic that might actually extend out over 100 years. And this is another picture that's showing you the same kind of thing. Here is a time scale from at the beginning, and then later on, and then late in this cycle of decomposition. So again, showing you soluble compounds like sugars and amino acids are breaking down initially. Then we're going to have the cellulose and the hemicellulose breaking down, and then we're going to have the lignin and maybe some of the waxes and other things that are degrading. Now look what's going on with biological succession. We start out with mixing and comminution, which is that shredding, soil organisms, animals using it, and also some of the microbes like the rapidly growing fungi and some of the bacteria are important. And then we move into organisms that are more specialized for breaking down things like cellulose and hemicellulose. And then we move into these really kick-ass fungi that are the ones that are needed to break down things as complex as lignins. There's other things that happen as well in the chemical composition of the organic matter over time. We have increasing in these carboxyl groups, decreasing in hydroxyl groups. The carbon to nitrogen ratio is going to approach about 10 and sort of hang out there, poise. They there. You're going to have more and more microbes, microbial products, like from dead microbes being created, and more formation of humus. So a lot of succession going on in this decomposition of plant material. The kinetics of decomposition, you remember this from our kinetics lecture, is often described by a single first order equation, but usually mixed equations do a better job. Remember, we talked about the double first order equation, using two of them to do a very good job of describing plant material decomposition, where you have a fast initial rate and a slower second rate. In this case, this graph shows you a combination of first order, a fast first order rate, and then a slow zero order rate. Look at its CO2 evolved this time rather than something disappearing over time. Remember, we measure products as well as parent material in following decomposition. And so you get this fit of this double rate kinetics equation with a first order and then a zero order rate. Cool, huh? You can totally relate to it. You could build this. And that rate of decomposition depends on different factors. Partly it's the biochemical makeup of the plants. You can see here's a branch compared to, say, a leaf of a deciduous tree. And the branch is much, much more slow to break down because it has really high lignin content and a high carbon to nitrogen ratio. So these two factors do a pretty good job of estimating how degradable a particular plant material is. Temperature and moisture are really important as well. You remember from the environmental factors lecture that with every 10-degree increase in Celsius temperature, what happens with the rate? Q10 doubles, not a bad estimate, although of course Q10 does vary, as we are finding out as we're looking at decomposition of things as the planet warms. But more or less a doubling with rate often. So temperature is really important, and also moisture content. And it's important on either side of the continuum. In really dry situations, there's not enough water to support life, and there isn't going to be much decomposition. And at the other end of the continuum, if the soil is super moist, then there's not going to be much oxygen present, and decomposition is going to be much slower. And we see that in bogs and places like that. Also, pH can be important, and whether or not the particular kind of organism is present, it's not really clear how important that is. It seems that there's usually organisms present that will decompose organic material that's present, but we now have new ways of looking at what particular kinds of organisms are around with these molecular techniques, and so we're learning new things about them, and we'll see. I think there's a lot we're going to learn as to whether there's particular kinds of organisms that are associated with faster rates of decomposition. Okay, and this is just, again, you can see I'm trying to connect you back to all the previous lectures. Do you remember this figure, this all roads lead to central metabolic pathways? And remember that we have all these complex substrates that are out in the environment. We've just been talking about them, like cellulose, starches, lignin, fats, and they break down through these preparatory extracellular type reactions and produce these simple compounds that are these central intermediate compounds that feed into the central metabolic pathways. And then you can follow how these organisms are able to build cells and make energy from all these different sort of molecules, some of them quite complex, bigger than their heads, requiring extracellular enzymes to be able to get them to a place where they can get into the cell and get to work. Okay, cellulose is one of the these important components of plant residues, the most abundant carbon input to soil, made up of glucose units. So they're repeating glucose units held together by the same kind of bond. And This is a great picture that shows you a cross-section of a cell wall, that then you can see this bundle of cellulose microfibrils making up that cell wall. And then if you blow up that microfibril, you will see that it's made up of many, many individual cellulose chains that are bundled up, and each one of them being composed of these repeating, boring same units of glucose that require this very specialized enzyme complex that includes an endo and exoglucanase, which are enzymes that cut either at the end of these at the cellulose units, or it can cut in the middle somewhere and break them apart into big chunks, but that are separate from the long chain polymer, and also cellobiose. Organisms that have these kinds of enzymes include primarily fungi, but there are some bacteria as well that can carry out these reactions. So it's fairly specific. Not all organisms can do it, but it's spread over, the ability to break down cellulose is spread over a broad group of organisms. And animals, of course, like cows, ruminants, can also break down cellulose, but they're not really breaking down cellulose, are they? We know what they're doing. They're hosting microorganisms in their complex, multiple stomachs that are actually the agents behind breaking down the cellulose into sugars and into single cell organisms, right, that the cow absorbs and uses for its sustenance. And so cellulose can break down under both aerobic and anaerobic conditions. That cow gut is certainly anaerobic. And the soil, the rate of decomposition of cellulose is going to be faster in aerobic conditions. Hemicellulose and chitin are two other forms of polysaccharides that come from plant materials and enter soil. Well, chitin doesn't come from plant materials. Hemicellulose is, unlike cellulose, not these repeating same sugar units. It's, in the case of hemicellulose, you're going to have different kinds of sugars, glucose, mannose, galactose, and they're going to be held together by different kinds of bonds. So it requires a variety of enzymes to depolymerize this hemicellulose, and usually it works better if you've got a variety of organisms that are working on it at the same time. Chitin is really interesting. It's a constituent of fungal cell walls and also insects, and it's rather attractive because it has nitrogen in it. So often organisms will go after it also to get nitrogen, and it has glucosamine units, again requiring a variety of enzymes to depolymerize it. It's not as systematic as cellulose as well. There's also starches and pectin that are constituents of plant materials. But you're kind of getting the point. We're talking about these large polymers that require extracellular enzymes to break them down into small enough units, which are going to be sugars, which can then be taken up into cells. In contrast, we have lignin. Lignin is this completely different beast, which is a very, very complex molecule that you can't even draw. It's so diverse, but you can see that it's got a lot of aromatic rings in it. It also has things hanging off those aromatic rings, aldehydes and hydroxyls and carboxyls and phenols, and that make it much more complex. And then you have some very, very recalcitrant bonds like ether linkages that are holding together these different constituents that make up lignin. And lignin is also has like a 3D structure, so it's challenging also to like break into it. Of course, in plant material, we're going to have lignin commingled with cellulose and hemicellulose, so we're never talking about one purely. The organisms that can break down lignin include brown rot and soft rot. I didn't put that here, and white rot fungi, and we'll talk about them in a minute. But one important thing is that the general metabolites that are formed when lignin is broken down are these variety of different kinds of phenylpropane units, which are aromatic rings with things hanging off of them. Some of them quite tasty or smelly, like vanillic acid, that are then broken down by other organisms that crack open the rings and create, again, intermediates for the central metabolic pathways in cells. So lignin degradation is carried out by brown rot and soft rot. They're partial degraders. I call them nibblers. They're more likely going to just sort of pick around the edges at some of these side chains and modify the overall lignin molecule, but not necessarily really breaking apart this lignin. So a lot of it is going to be left behind. The brown rot leave wood looking like this, where they are using the cellulose and modifying the lignin a little bit, but leaving a lot of lignin behind as this brown, and you can see it in this brown color that's really brittle. You've seen this when you've walked in the woods. And then soft rot fungi also use cellulose and are actually able to handle a broader set of conditions than the brown rot and the white rot. For example, wetter conditions, and you see this sometimes in trees that are have been colonized by soft rot, that they're really mushy, and you can look at this Wikipedia and it'll give you an overview of these organisms. So different kinds of fungi are carrying out these reactions. And then we have the white rot fungus. So unlike the nibblers, it's a blaster. It's this phenomenal group of organisms that can depolymerize the lignin and completely break it into smaller bits, often leaving a white cellulose behind, as you can see in these pictures, and also leaving behind mycelia as well, and hence the name white rot. And so they use these heavily oxidative, indirect, random, promiscuous enzyme systems that, unlike that sort of lock and key that we saw with the cellulose decomposition, they're just spewing these enzymes in all directions with no particular target, and just letting them hit whatever they come into contact with. And what is unique about it is that they help generate free radicals. Remember the toxic condition of an aerobic environment. Well, they will actually catalyze the generation of more of those that then will combine with the enzymes that they release, that are what creates the power of breaking apart this really recalcitrant, difficult to decompose lignin material. And this picture shows it in more gory detail, at least for one particular kind of white rot fungus, but I'll let you look at that at your own leisure. And this is interesting because it happens during secondary metabolism. Do you remember that when we were talking about growth kinetics, that after the rapid phase of growth and before the death phase, when we're talking about growth of populations, there's this period of secondary metabolism, stationary growth, and the white rot lignin decomposition enzyme system is active in that during secondary metabolism. It's a kind of secondary metabolism. Some people think it's turned on when the fungus runs out of other things to eat, and then it decides to go after the lignin, and so this enzyme system is induced. So what's interesting about it is because this enzyme system is so non-selective and can break down anything you put in the path of it, humans have learned to put all sorts of nasty pollutants in the path of these white rot fungi. And indeed, if they're actively decomposing this wood material and producing all these enzymes. Those enzymes will also work on the pollutants that you put in their path, that you combine with it. And this works really well in controlled conditions like shown down here, where you keep the fungi really happy, keep the conditions really good, have them essentially working off of wood and trickling the pollutants actually ideally through the wood in a solution, passing over them. And in such kind of controlled bioreactors, you can get really good decomposition of some of the most difficult to decompose compounds like PCBs and dioxins, which are really hard to biodegrade, but can degrade in this lignin system. Unfortunately, when you try to inoculate like contaminated soil with white rot fungi, they, I think, look up at you and say, what are you doing putting me here in a soil environment when I am a specialist on wood? And I'm not going to do very well here. And in fact, if you inoculate the soil with chips of wood with the fungus on it, they tend to just stay on the wood and don't move into the soil and break down the pollutant in the contaminated soil. So it's difficult to get this to work as a soil remediation technique because they really are out of their environment in that case. Okay, now we're going to talk about B, the second path, which is after the breakdown of the plant material, is the creation of soil organic matter. Okay, and I have to first give you a warning that this humification used to mean more than it means anymore. We talk about humus as a name for soil organic matter as a final product, not this kind of humus, but also not this kind of humus, which is created. Traditionally, we used to believe that soil organic matter was made up of humic substances, which included fulvic and humic acid and humin, the material left behind. And we now know that these different pools are complete laboratory artifacts that were just results of how we processed our soil samples. We'd bring them in and we'd put harsh acid on them, then harsh alkali on them, and essentially created compounds that did not exist in the soil through just our handling of it, and led us on wild goose chases and thinking that soil organic matter had very different properties from what it actually does have. We now know, using non-invasive methods, many of them borrowed from medicine, like from imaging the body. We've been imaging soil organic matter, and now we can look at it without destroying it and creating these artifacts. And we've learned a variety of new things about it. So anyway, how is soil organic matter formed? Again, there's two stages in it. One are these preparatory reactions where we're breaking down plant residues into smaller constituents, and second, creating microbial cell wall material that is really important as a precursor of soil organic matter. And then we have stabilization processes, so we'll talk about that. But first of all, this was a big discovery not that many years ago, that a very large portion of soil organic matter actually comes from dead microbes. Of course, the microbes ate the plant residues that went into the soil, but they laundered it. And so that plant material, a large portion of the plant material, doesn't go directly into soil organic matter. It is taken up by microbes and they die, and that's what makes the soil organic matter. And this is a picture showing you a hyphomicrobium growing on a surface, and you can see fragments of different soil organic material and microparticulate matrix. I can't actually describe this picture to you very well, but I think one way of looking at it is microbes are the eye of the needle through which the carbon flows into soil organic matter. right? Before we thought that the plant material just flowed right into the soil organic matter, but a lot of it is going through microbes first. Very important discovery. Okay, stabilization now. So we used to think that the plant material broke down into a lot of smaller pieces, and then there were condensation reactions that were carried out by extracellular enzymes of fungi that piece these pieces back together into this complex material known as soil organic matter, and that the structure of the plant material was seen to be very important in how long that organic matter would last. But now we know it doesn't really work that way, or maybe there's a little bit of that still going on. But the emerging understanding of what's going on is indeed you have this plant material that's breaking down, but reminding us that it's coming not just from the surface, from the leaves, but also from roots and from the rhizosphere and from the microbes being made. They're all sources to soil organic matter, as well as fire. The importance of fire is being increasingly recognized. The charcoal that we find in soil is also important in making soil organic matter. But anyway, these materials then enter in the soil, and they are stabilized in the soil by different processes. So one is going to be sorption of the organic matter from these different sources to clay or oxides in the soil, where they are going to be made unavailable to microorganisms through these associations that are going to be created between the organic and the mineral components. Also, physical disconnection, where some of that organic matter is going to be trapped inside soil aggregates in particular. And there's a complex body of science that looks at creation of aggregates, and we've touched on it somewhat. But when you get down to the scale of the organic matter and the minerals and the microbes, there's been a lot of work on really trying to understand what is going on at that scale. That's quite interesting. So that's another way that it's stabilized, right? Is it's physically protected and removed from where microbes and enzymes can get at it. And then freeze, thawing, and wet dry cycles are really important as sort of a driving force, and perhaps tillage too. Some amount of tillage seems to be good for creating organic matter. Too much tillage obviously is going to not be good. It's going to actually lead to decrease in soil organic matter, but a certain amount of mixing through freeze, thaw, wet dry, or tillage is going to bring together the organic matter with the clay particles, or help form aggregates that will then protect the organic matter. And all right, I forgot to do all that. Okay, and this is showing the clay physically protecting the organic matter. Here's a big chunk of organic matter and all these different bonds that are going to be holding the organic matter to clay and making it unavailable, including really weak bonds like hydrogen and van der Waals, and also some more complex kinds of associations. And then, which leads me to think that we shouldn't really be calling it organic matter. We should be calling it organo-mineral matter, because really the preserved stuff is going to be very strongly associated with the mineral portion of the soil, be it just through this adsorption to clay, or be it inside aggregates. And here's a picture just showing you the physical disconnection. The organic matter becomes sequestered inside aggregates, and this is a pretty complex figure from Johann 6, showing the different mechanisms for how the carbon gets trapped inside aggregates. And you know from before we looked at those figures of how soil aggregates are made, and you saw there how the carbon gets inside and starts to build or create these nuclei for aggregates to grow around them. And there's the extracellular polymers as well that microbes produce. And then you can have roots that are moving inside aggregates as well that are important, and fauna are going to also move material into aggregates, and then the mineral reactions that we were talking about just before in the slide before are also important in creating these figure, but that's just to remind you about what's going on with aggregates. So what we have at then any one point after soil's been around a while are these different pools of soil organic matter that have these different ages that are associated with how rapidly they're going to turn over. So we have really rapidly turning over organic matter that's like the fresh plant residue, which is only going to have a turnover time last for about a year or less. It's going to be decomposing pretty fast. We have the microbial biomass, which is less than 5% of the soil organic carbon, but turning over quite rapidly, turning over every year, and therefore releasing nutrients that are tied up in it, and releasing carbon tied up in it. So even though it's a small pool, it's turning over so fast that it may actually release a lot more carbon than a bigger but slower turning over pool of organic matter. And remember, fumigation, extraction, and the methods lecture, right, is a method that we use to measure nutrients that are associated with this microbial biomass, be it carbon, be it nitrogen, being phosphorus. Then we have an active fraction, which has a turnover time of 15 to 100 years. This is going to be some of the organic matter that will be a slow release of nutrients, but will take a while for those nutrients to become available. And then we have stabilized organic matter, which is much, much, much older, very stable, important for soil structure, that can be quite old. Let's not forget that we also have inorganic carbon. I used to think this was totally irrelevant. I used to work on soils of pH 4 in upstate New York, which had very little carbonates in them. But here out in California and a lot of soils in the West, the carbonate content can be quite important. In fact, can be a big portion of the total carbon in soil if the organic matter content is low and is high pH. So let's not completely forget about those. Okay, so what are some of the factors affecting soil organic matter accumulation and turnover? So quantity of carbon is going to be important. The more organic matter that you put in, the more soil organic matter that's going to come of it. And if you reduce the amount of crop residue that goes into the soil, like you burn the residues or you take a lot off to make biofuels, for example, right, it's going to lead to less accumulation of soil organic matter. The quality of the carbon is important, and people are thinking now that you can't have too recalcitrant of organic material that you're adding to soil. You need some of it to decompose, particularly to make microbes to be able to make soil organic matter content. Disturbance is important. Erosion obviously is going to just move your soil off the surface and where a lot of the organic matter is, and just physically remove it from the system. Plant removal is going to be important. Tillage, too much tillage is going to introduce oxygen and expose protected organic matter, and that will lead to less soil organic matter. But remember, I said a little bit of tillage might be important for bringing the actors together and helping create soil organic matter. Temperature, lower temperature is going to lead to greater soil organic matter creation, particularly if there's enough plant productivity going on, because the lower temperature is going to slow down decomposition rates. And high moisture content environments lead to greater accumulation of soil organic matter. And this is something that's of concern up in the Arctic, where there's a lot of a lot of soil organic matter that is been accumulated in a low temperature and high moisture content environments like in bogs, where now with warming and some drying out of those environments, a lot of that carbon may now be available for decomposition. Low moisture environments also tend to have low organic matter content. This is like a desert, largely because there's low plant productivity and low inputs going into the soil. Texture is very, very important, as you know from before. Higher clay soils tend to accumulate more soil organic matter. If you want to sequester carbon in soil, go for higher clay soils rather than sandy soils. And then again, microbial communities, does it matter? Like if you have more fungi in your community that have higher carbon to nitrogen ratios, meaning that they would bring more carbon into their tissues and hopefully makes more soil organic matter, does that influence how much soil organic matter is going to accumulate in that soil? And people are doing research on that. This is a complex model showing you the flow of carbon from plant material or either surface or roots through the different pools in the soil. And here's a picture showing you a soil with a lot of organic matter compared to one with less, showing the difference in the color. Okay, agricultural practices change soil organic matter, and we've had huge losses of soil organic matter in soil throughout the world as we've started to grow food. And this diagram shows you soil organic matter in mass over time, with time since cultivation started, and looking at these different pools of soil organic matter from essentially plant residues on the surface to active, rapidly turning over organic matter, a slow pool and a passive pool. And you can see over time, like before cultivation begins, you have this nice mix of a lot of plant residues on the surface and a good amount of active organic matter and a lot of slow organic matter. So these more rapidly turning over pools are rather large, but as you start to farm with tillage, with removing organic material from the plots of land, with reducing biodiversity, all the different things that we do in agriculture, you're going to have losses of these different pools, particularly of these more rapidly degrading pools. Notice that the passive organic matter fraction is not that sensitive to these practices associated with agriculture, and it's not going to decline as much. But you can get to a point where you have very little material duff on the surface anymore of the soil, not much active material, and very small pools of slow organic matter. And this is the situation throughout California. California has experienced tremendous losses, like 50% loss of soil organic matter is not uncommon in California, and also throughout the world. And some of the CO2 that we now have in our atmosphere comes from this. But the good news is that we can build some of that soil organic matter back up. People who are interested in carbon sequestration are certainly interested in this, but also just for the purposes of soil health, it's really important to get that soil organic matter back. And that can be done through intentionally growing plants like cover crops that go back into the soil, or not removing as much residue from the soil, or adding external inputs of carbon like compost and manure. The role of soil organic matter is many. There's biological functions that include providing energy to all the microbes that do all the things in soil, releasing nutrients associated with them, often slow release that are available to plants over time, contributing to the overall resilience of soils. Physical functions include improving the structural stability of soils, water retention. Higher organic matter means the soil is going to hold more water against drought, We've seen that with organic farms that did much better in the Midwest during the drought that they had some years ago than did conventional farms that had lower organic matter content in their soils. And also changing the thermal properties of soil. Darker soil that's associated with higher organic matter is going to warm up faster and retain heat. And chemical functions, increased cation exchange capacity with high organic matter, greater ability to buffer changes in pH, and ability to create complexes, that meaning retaining cations in the soil through other forms than just cation exchange capacity. And here's a big list of other things, some of them the same. It's a little bit redundant, but some other aspects of soil organic matter, including some people have concerns about too much soil organic matter, particularly like plant residues lying on the surface, may be associated with harboring some diseases or pests. This was a concern for the rice farmers who used to burn their rice straw every year. They were doing that when I first came to California 26 years ago, but it created this enormous air pollution problem with all the silica flying around in the air during that period and terrible air quality. And so now actually the rice straw is allowed to decompose in the soil. Oh, I forgot to tell you, the rice growers felt they had to burn it because there's a disease called take-all, I think, that they felt like burning was necessary to break the cycle and prevent it from infecting the rice. But it turned out that just decomposition of the rice straw under like flooded conditions during the winter was also sufficient in breaking the cycle of that disease. And so now burning is not done anymore, and the organic matter degrades and goes back into the soil. This is a very good article by Tom Philpott. If you don't know about him, he's a wonderful journalist who writes a lot about soils and agriculture, and this is a really nice article about soil organic matter in a context of organic agriculture. Okay, we're done. Some supplemental fun. Compost is accelerated human-controlled decomposition. The same kinds of things that we saw in looking at the soil are happening faster on crack in preparation of compost, except compost does not have the mineral component. So we're not making that complex material known as soil organic matter in composting, but we are having that condensation reaction where the components from the breaking down plant material are formed into these complex organic compounds, these big molecules. Okay, Zai pits is this amazing practice in Africa, in Niger, and other areas in the Sahel, where organic matter inputs are concentrated into these localized areas. This is a more complex rendering of it, but you focus on putting organic matter and a seed and maybe manure and water into these like spots, into these essentially nucleating agents that then create these islands of fertility and help to actually spread the growth of the plant from radiating out from these spots, leading to slow but constant revegetation of these areas that were originally extremely arid without plant material, but are now seem to be recovering well where these practices are implemented. Check it out. It's really interesting. Zai Pits is an amazing farmer who is responsible for this in this area. Bog people. Bog people are humans that were put in in bogs, and there's this process that occurs, the Maillard reaction, under anaerobic conditions in bogs at low pH that lead to conversion of the organic matter in these humans and in their clothing and the ropes around their necks that make that organic material very recalcitrant, unavailable to microbes. Hence they are preserved, and people dig them up and make stories about what went on and why are they there. And don't forget methane. Methane is another important part of the carbon cycle. It's the littlest carbon compound, just one carbon. And we talked about it plenty, I think, already, but I'm just reminding you that do think about methane as well. And remember, it's a potent greenhouse gas. And finally, soil organic matter is critical for soil health. And this is a very nice YouTube that you can watch that talks about soil organic matter and its importance in soil health. If anything is necessary to increase the health of a soil, it's increasing the soil organic matter. And that is a common agreement on that. So very important area. And the figure below is just for fun. Okay, that's it.